So what's the clinical relevance of all this? So we did a study in San Francisco General Hospital, which is a public hospital, and we looked at gram-negative bacterial isolates from bloodstream infections, okay? And we looked for drug resistance integrons, which are mobile elements that carry drug resistance genes. We looked for specific drug resistant gene cassettes on the integrons, and we specifically looked for CTXN gene. And here we found that most of the bloodstream isolates, gram-negative bacterial isolates, were E. coli, which is true all over the world. And there are about 50% were resistant to at least one drug. And we found these class 1 integrons and the gene cassettes in the integrons, aminoglycoside aminotransferase gene, hydrofolate reductase gene, etc. So these are all found in the E. coli isolates. And we found this particular cassette configuration, DFAR17AD, AADA1, and AADA2 cassettes in 100% of 41 of all BSI isolates from San Francisco General Hospital. CTXN15 was found in 15 of the E. coli isolates. Okay? So again, this is what I told you, right? This is the most common ESBL gene. And then we did we compare the sequences to the NCBI database. Look what you see here. Okay, this particular sequence, 100% identical sequence, found in all these different organisms, not just in gram-negatives, but even in staph, which is a gram-positive organism. And they were found from Australia, China, Russia, Vietnam, Portugal, Japan, Mexico. Okay? And not just from humans, but from cow, respiratory device, catfish cultivation, food microbiology, pork intestines, etc. So now, here's evidence that this particular gene is found all over the world, not just from humans, but from all kinds of sources. Okay? So, how it's distributed. And one of the interesting things we observed was that, so CDC, this is the CDC definition, so if the blood culture becomes positive within 48 hours after admission, that's considered to be community-acquired infection. Okay? If the blood culture becomes positive after 48 hours, that's defined as being a hospital-acquired infection. So if you look at this, look at the E. coli. 78% became positive, culture became positive in less than 48 hours. So that means all these bloodstream infections with E. coli were community-acquired. Okay, not in the hospital. Whereas Pseudomonas is mostly hospital-acquired. Okay? So this means that if this hospital stopped using all antibiotics right now, it will not prevent E. coli, drug-resistant E. coli infection. It will have no effect. Okay? Because they're acquired outside the hospital. So, in all your hospitals, Brazil and Africa, all these other countries, you are told to decrease the use of your antibiotics in the hospitals, right? Okay? That's important. Of course, you have to do that. But it's not going to be effective for drug-resistant E. coli. Okay? You have to do something else. What do you have to do? Stop using antibiotics in animals. Food animals. Huh? In the clinics? Oh, yeah, it's possible. But as I showed you, the UTI in Berkeley, there was no effect in increasing prevalence over a 6-year period. Okay? And how do you explain clone of E. coli that's coming into Berkeley and all these other places? It's not the clinical use of antibiotics. It's a clone that happens to be resistant to spread. That's what contributes to the increased prevalence. It's not the use of antibiotics in the humans. It's the use of antibiotics in the animal reservoir, and then the spread of these drug-resistant clones that ultimately determine the prevalence of drug-resistant infections in the human population. Okay? It's a different concept. And this concept will not become clear until you do the molecular biology work, analysis. Okay? That's why you do molecular epidemiology. So, this was a question we asked. What proportion of clinically relevant healthcare-associated infections are caused by bacteria that harbor drug-resistant genes that originate food microbiota? Answer is 38%, at least in the San Francisco study. So 63 of 167 drug-resistant bloodstream infection E. coli sets from San Francisco General Hospital during the study period carried identical drug-resistant genes, these genes, that are also found in the food microbiota. Okay? And this is probably an underestimate, because we only looked at these genes. If we look for other genes, we may find more. Okay? So I think it's probably more than 50%. So, why do we see this? I think this is the reason. Okay? So worldwide, this is just a 1 month of air travel in June of some year, I forget which year. But people are just moving all over the world. Food is being distributed all over the world. And this is the new world. Okay? And so, we have to really sort of understand this type of the epidemiology of drug-resistant infections. It's not what we used to think about. And so, just to summarize, mobile drug-resistant genes with identical sequences are widespread across different gram-negative bacterial strains, species, isolated from patients with BSI. The same mobile drug-resistant genes are found in bacterial isolates in different regions of the world and from both clinical and non-clinical sources. And use of antibiotics in hospitals appears to have limited impact on the selection of drug-resistant E. coli, BSI, in hospitals. So, you can just look at the references. These are all published. You can just study them. And I will stop here.